Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad MSc Entrance Examination conducted for the subject Plant Biology and Biotechnology in the year 2012. So coming to the first question, what are the essential elements that are bound to chlorophyll, heme and plastocyanin respectively? So they are Mg, Fe and Cu. So the answer is option A, magnesium, iron and copper. Then plants use fats or lipids mainly for energy storage. Option B. The commonly used reporter gene encoding green fluorescent protein or GFP was isolated from Aquaria victoria. Answer is option B. A silent mutation is one that replaces an amino acid with an equivalent amino acid in a polypeptide. So answer is option B, replaces an amino acid with an equivalent amino acid. Then next one, the best choice for producing plants that are homozygous for all trait is. So the answer is option C, that is anther and pollen culture. Which one of the following is unique to gymnosperm leaves? So it is the endodermis that is unique to gymnosperm leaves. The long cylindrical needle leaves of pine, that is a gymnosperm, have an outer chlorenchyma made up of lobed cells and a central vascular bundle. The two tissues are separated by a boundary of parenchyma known as endodermis. So the answer is option B. Then next one, in a typical standard curve, which one of the following is generally represented on the x-axis? So on the x-axis we have the concentration of the chemical tested. Now if you are doing Fallin-Lowry's, you will have concentration of the protein tested. Different concentration of proteins you will be plotting on the x-axis. So answer is option B. Then the expression of tryptophan operon in E. coli is regulated by the availability of the amino acid tryptophan. This regulatory process is called as attenuation. Answer is option C. The next one, a non-coding RNA molecule which is hardly 400 nucleotides in length, circular in nature and highly infectious to plants alone is called as viroid. Answer is option C. Plasmids are inherited stably during cell division in bacteria, but they are not always equally apportioned into daughter cells and sometimes are lost. This loss of plasmids can occur spontaneously or be induced by treatments that inhibit plasmid replication but not host cell reproduction. The loss of plasmid is referred to as curing. That is, plasmids are stable during cell division in bacteria, but they are not always equally divided into both the daughter cells, and sometimes they may be lost during cell division. And this loss of plasmid can occur either spontaneously or be induced by treatments that inhibit plasmid replication but not host cell reproduction. So if we treat a particular bacterial cell with specific chemical that will inhibit plasmid replication but not the host cell reproduction, then it can result in loss of plasmid in the next generation. So this loss of plasmid is referred to as curing of plasmids. Answer is option B. Then Pseudomonas is one of the most important genera in the family Pseudomonadaceae. Some of the members of this genus called fluorescent pseudomonads show fluorescence. We detect this phenomenon of fluorescence by such pseudomonads by noting observations under. So the members of the genus Pseudomonadaceae, they show fluorescence. Okay, Pseudomonas is one such. So we can detect the fluorescence produced by these pseudomonads by observing under. So by observing under fluorescent microscope, you can detect fluorescence. So answer is option A. What is the correct path for water right from the time of absorption to transpiration in a plant? So transpiration is the evaporation of water at the surface of the spongy mesophyll cells in leaves, followed by loss of water vapor through stomata. So that is transpiration. So we have to write the correct pathway of water right from the absorption. So absorption take place at the root hairs till transpiration. So the answer here is option C, that is root hair, xylem, spongy mesophyll, and again xylem, and then stoma. So answer is option C. Antibodies are produced in transgenic plants expressing the antibody genes of a mouse are called. So they are called as plantibodies because they are produced in transgenic plants. So they are called as antibodies produced in plants through transformation techniques are called as plantibodies. Then Dicathium, Cynodon, Digitaria are example of producers in. So they are example of producers in the grassland ecosystem. So answer is option A. Then Sanger DNA sequencing, site-directed mutagenesis and PCR are three important techniques routinely used in modern biology. Out of the common reaction shared by these techniques, pick the odd man out. So single-stranded DNA template. For DNA sequencing, PCR, every site-directed mutagenesis, you require a single-stranded DNA template. Then DNA primer complementary to the template. Of course, you require a primer for all the three techniques. Then annealing of the primer to the template. So for all the three also, you require the primer be annealed to the complementary bases in the template. Then initiation of reverse transcription. So that is not commonly shared by all the three techniques. So option D. Then an amphidiploid species was produced by making cross between Brassica oleracea and Brassica nigra, followed by chromosome doubling of the hybrid. What is the chromosome number of the amphidiploid? So they have given 2n is equal to 18 plus 16. You will get 34. So that is the chromosome number of the amphidiploid. So answer is option C, that is 34. Then which of the following organism can be used for mapping the loci with respect to centromere? So Chlamydomonas reinhardti can be used for mapping the loci with respect to the centromere. So answer is option C. Then next one, capsid assay. So capsid assay chemical modification at the 5' end of a eukaryotic mRNA molecule, that is a cap, mRNA capping. DNA binding site for catabolic activator protein, that is CAP region, not capsid. Protein coat that surrounds the DNA or RNA genome of virus, that is more correct, that is a capsid. Name of high insert capacity vector which has the characteristic both plasmid and cosmid, that is also not correct. So answer is option C. Then insectivorous plants evolved to combat the deficiency of dash in the soil. So it is the deficiency of nitrogen in the soil. So answer is option D. Then next one. When 30% of chiasmata are formed between two loci X and Y among the tetrads of an individual of the genotype capital X small x capital Y, the percent of gametes expected to be capital X small yr. So they have asked for the parental combination of the gametes capital XY. So chiasma, that is percentage, is 2 into crossover percentage, or crossover percentage is equal to half of the chiasma percentage. So these are the XY, these are the parentals, and small x capital Y, they are the parental. They will produce 4 types of gametes, 2 of which will be recombinants. So that is 30% of the chiasmata represent the crossover percentage. That is half of this chiasma percentage will represent the crossover percentage of the gamete. That is those which are recombinants. So it will be 15% and 15% for the crossover percent or the recombinant gametes. And out of this will be 35% each will be the parental combinations, that is capital X small y. They have asked for the percentage of gametes expected to be capital X small y. So the answer will be 35%. But the answer is not seen in any of the options. So the one which is near to 35% is 30%. So I think the answer will be 30% as per me. I am not sure. If you have any problem with my calculation, you can comment it. Then cytoplasmic male sterility in maize has been exploited for hybrid seed production. This trait is transmitted by the seed parent. Cytoplasmic male sterile lines are maintained by. So cytoplasmic male sterile lines can be maintained by repeated crossing to a sister line, also known as maintainer line. And this maintainer line is genetically identical except that it possesses normal cytoplasm, that is male fertile. So answer here is crossing with the genotype same as the cytoplasmic male sterile line. So answer is option D. Banana bunchy top, citrus exocortis, witch's broom, disease of brinjal are caused by which of the following respectively? So banana bunchy top is caused by virus, citrus exocortis is caused by a viroid, and witch's broom is a phytoplasma. So the answer is option A, that is virus, viroid and phytoplasma. Dash and dash are cause plant overgrowth due to increased cell division and abnormal cell enlargement respectively. So hyperplasia is the enlargement of the plant tissue due to excessive increase in the number of plant cells produced. Then hypertrophy is excessive growth due to enlargement of individual cells. Hydropathy is our therapeutic system that professes to cure all diseases with water. And homothallism having male and female reproductive structures on the same thallus. And heterothallism is the production of two types of thallus or hyphae in fungi designated as plus and minus type. These hyphae undergo nuclear fusion to produce the zygote. So here they have asked for dash and dash cause plant overgrowth due to increased cell division and abnormal cell enlargement respectively. So it is hyperplasia and hypertrophy. So hyperplasia, as I have told earlier, is the enlargement of plant tissue due to excessive increase in the number of plant cells produced, and hypertrophy is excessive growth due to enlargement of individual cells. So answer is option A, that is hyperplasia and hypertrophy. Then of the following light sources, only dash light is capable of retarding plant growth. So as per me, the answer is yellow and green, because yellow light have lowest effect on plant growth. So I think the answer is yellow and green. I am not sure because I don't have the answer key with me first of all. So after reference I have found that yellow and green might be the answer. If you have any comments, please write it in the comment section. Then leguminous plants exhibit biological nitrogen fixation in which bacteria convert nitrogen to ammonia. Then next question. The effect of light on seed germination is called as photoblastism. Phototropism is the orientation of plant or other organism in response to light. Then photoperiodism is the response to changes in day length that enables plants to adapt to seasonal changes in their environment. And phototaxis is bodily movement of a motile organism in response to light. So they have asked for effect of light on seed germination, and it is called as photoblastism. Then next one. Plant exhibits vivipary when they are deficient in. So vivipary occurs when seeds or embryos begin to develop before they detach from the plant. So it occurs during when there is deficiency in abscisic acid. So answer is option A. Then myoglobin and the subunits of hemoglobin they have very similar tertiary structure but different primary structure. Answer is option D. Root hairs are found in the region of the root called the region of maturation. Answer is option D. Which one of the following statement is correct? Mutation are errors in the DNA that are always harmful. No, there are silent mutations also which are not that much harmful. Then mutation occurs in the presence of carcinogens. Need not be. Thymine dimers occur as a result of ionizing radiations. Then mutations increase tumor growth. Need not be. Then mutation occurs spontaneously at a low rate. So which of the statement is correct of mutation is option D. What is essential element needed to make ATP? So it is phosphorus, adenosine triphosphate. So answer is option B. Which one of the following statement is true? C4 plants don't have a rubisco enzyme. Then very few plant cells have mitochondria. Amylases break down starch into sugars. Starch synthase is an enzyme, is the name of a plant enzyme that synthesizes starch. So C4 plants don't have rubisco enzyme. That is not true, because C4 plants rubisco occurs in bundle sheath cells, whereas in C3 plants it is in the mesophyll cells. Then very few plant cells have mitochondria. That is also not true. Mitochondria is present in most of the plant cells. Then amylases break down starch into sugars. That is correct. So R is correct. And starch synthase is the name of the plant enzyme that synthesizes starch. So S is also correct. So answer is R and S. Option C. How do sclerid differ from sclerenchyma fiber? So the answer here is sclerids are short branch, whereas fibers are long and narrow. Answer is option D. Then in option C, sclerids only have bordered pits, whereas fibers only have simple pits. So that is not correct, because sclerids have simple pits. Next one. A sequence of amino acid in a certain protein is found to be serine glycine proline glycine. The sequence is most probably part of a beta turn. It cannot be any alpha helix, because glycine and proline are helix disrupting proteins, amino acids. So alpha helix it cannot be, neither can be anti-parallel beta sheet and parallel beta sheet, and this combination, that is serine glycine proline glycine, occurs usually in the beta turns. So answer is option D. Then which of the following plant hormones can induce parthenocarpy in many plants? So So it is auxin that can induce parthenocarpy in many plants. So answer is option B. Then next one. A cell within a cell is found in. So the suspensor is a supporting structure during plant embryo development that pushes the embryo proper into the endosperm cavity and connects it to the surrounding maternal and endosperm tissue to facilitate the transfer of nutrients and plant hormones. Then the sieve tube is the conducting element of the phloem. It is formed from a vertical series of elongated cells interconnected by perforation in their walls in areas known as sieve plates. Then when the tapetal cells are polyploid and are specialized layer of nutritive cells found within the anther of flowering plants. So pollen grains, as per me the answer might be pollen grain, because when a pollen grain is shed at the 3-celled stage, it contains a vegetative cell and a 2 male gametes. So as per me it might be pollen grain. If you have any problem with my answer, you can give it in the comment section. So answer is option C. Then next one. Which functional group of the protein is identified by Biuret test? So it is the amino group. So answer is option B. Then next one. Polysaccharides can be formed from monosaccharides by the removal of. So they can be formed from monosaccharides by the removal of water. Answer is option A. Which of the following statement is true? Chlorophyll B is not essential for photosynthesis. Then chlorophyll B is essential for photosynthesis. Chlorophyll A is not essential for photosynthesis. Both chlorophyll A and B are essential for photosynthesis. Now chlorophyll B is an accessory pigment because it is not necessary for photosynthesis to occur. And all organisms that perform photosynthesis have chlorophyll A, but not all organisms contain chlorophyll B. And chlorophyll A is the most important pigment in photosynthesis. So the answer here is chlorophyll B is not essential for photosynthesis. Answer is option A. Then next one. Starting with mitosis, what are the other stages of the cell cycle in order in which they occur? So they have asked from starting from mitosis. So after mitosis is completed only by cytokinesis. So the first one will be cytokinesis. Then comes the G1, S and G2 phases. So answer will be option A. An infant is born with dwarfism. Which of the following statement is true about the parents? So neither is a carrier of dwarfism. Then one parent may be a carrier but the other may not be. Both must be dwarfs and both are carriers of dwarfism. So as per me, if both are dwarfs, then it is a recessive trait. They have given small t only in the question. So if both are dwarf means they will be having small t mother and small t father. So they will give rise to an infant with small t trait. So answer C is correct. But if both are carriers of dwarfism, then also you can get a dwarf child, because carriers of dwarfism means they will be heterozygous. So they will be capital T small t into capital T small t. So there is a chance that you will get a infant born with small t small t. So as per me, both C and D are correct. Then what is the method that can be used for predicting the three-dimensional structure of a protein based on known structures from one or more related proteins? So it is called as homology modeling. Then the frequency of a plant means frequency is the number of times a plant species occur in a given number of quadrants. So the answer is option D, the number of times the species is encountered in the community. So the answer will be option D. Then ferns cannot survive in dry area, and this is due to their underground rhizomes, small gametophyte, dominant sporophyte, swimming sperms. So the answer is option D, that is swimming sperms. The next one. In a tall tree, water can move from the soil to the top of the tree by using the following property of water. So osmosis, ionization, capillary rise, addition, cohesion of water molecules. Now capillary action helps bring water up into the roots, but capillary action cannot pull the water up a small distance, after which it cannot overcome gravity. To get water up to all branches of leaves, the force of adhesion and cohesion go to work in the plant's xylem to move water to the furthest leaf. So answer is option D, that is adhesion and cohesion of water molecules. The next one. When an E. coli is grown in a medium containing glucose and lactose, the Lac Operon is not expressed. This is because glucose interferes with. So low level of transcription, low level transcription of the Lac Operon occurs when glucose is present and lactose is present. The Lac repressor is released from the operator because the inducer lactose is present. Because lactose is present, the repressor will be removed from the operator. Then CAMP levels are low because glucose is present. Thus CAP remains inactive and cannot bind to DNA. So transcription only occurs at a low leaky level. So answer will be option B, that is binding of activator. Glucose interferes because they have asked for glucose. Glucose interferes with the binding of the activator. So answer is option B. Which of the following cells or tissues is not dead at functional maturity? So at functional maturity, colon-chyma is not dead. So answer is option B. The next one. Majority of bryophytes are smaller due to limitations in. So they are smaller due to limitations in water transport. Answer is option D. Then some of the halophilic microbes accumulate high concentrations of intracellular glycerol to counterbalance the osmotic pressure from the high level of extracellular salt. One such organism is used for microbial production of glycerol. Choose the organism that is used for glycerol production. So for glycerol production, Dunaniella salina is used. Answer is option C. Reverse transcriptase is an enzyme which converts an RNA molecule into a complementary cDNA molecule in the presence of some additives, is basically isolated from. So it is isolated from murine leukemia virus. So answer is option C. And E. coli deficient in RNA polymerase 1 would be, DNA polymerase 1 would be deficient in. So it will be deficient in repair mechanism because DNA polymerase 1 is required for repair. So answer is option C. The next one. Halophytes are special type of xerophilous plants which inhabit saline soil. They develop a special type of negatively geotrophic roots called. So halophytes are usually found in case of mangroves. Halophytes are usually plants which are seen growing in mangroves. Okay, and are special type of xerophilous plants that inhibit saline soil. So they develop special type of negatively geotrophic roots. They are called as pneumatophores or breathing roots. Answer is option B. Then next one. Who proposed the dual theory that lichens are made of algae and fungi? It was Simon Schwanner who proposed that lichens are made of algae and fungi. Then next one. The embryo in a seed give rise to the new. So the embryo in a seed give rise to the new sporophyte. Answer is option A. Periodontosis may lead to fall out of tooth out of its socket. Identifying the bacteria causing this condition. So it is caused by, that is periodontosis is caused by Porphyromonas gingivalis. So answer is option D. Then Ames test is used to. It is used to measure the mutagenic effect of various chemical compounds. So answer is option D. Then the term cyclosis refers to. So cyclosis refers to the movement of cytoplasm within eukaryotic cells. So the answer is option B, the movement of cytoplasm within eukaryotic cells. Then next one. Which of the following is the biodiversity hotspot of India? So 4 biodiversity hotspots are there in India, that is Himalayas, Indo-Burma region, Western Ghats, Sundaland. Okay, these are the 4 major hotspots in India, and Eastern Himalayas and Western Ghats is the answer. So answer is option C. Then buds that die on roots are referred to as. So they are referred to as adventitious buds. Option B. Then next one. How many different types of gametes an individual of genotype will produce? So they have given an genotype which is having 3 heterozygous genotypes, that is capital A small a, capital B small b, capital C small c. So we have the formula 2 raised to n, where n represent the number of heterozygotes in the given genotype. So there are 3 heterozygotes here, that is 1, 2 and 3, that is capital A small a, capital B small b, capital C small c. So 2 raised to 3, n is 3. So 2 into 2 into 2, that is 8. So the answer is option B. Which vector is constructed using F episomal factor of E. coli? So the vector is called phosmid vector. Answer is option C. A procedure that uses ultrasound to cause random breaks in DNA molecule is. So it is sonication that causes random breaks in DNA molecule. Answer is option B. The cell walls of this genus is distinctly different from other groups of bacteria by virtue of high amount of mycolic acid. So it is Mycobacterium. Answer is option B. So next one. In sweet pea plant, flowers are either purple or white, which are determined by 2 unlinked genes. The allele of the first gene are capital P and small p, and those of the second gene are capital W and small w. The plant must possess at least one capital P and W allele to bear purple flowers. The genotype lacking the dominant P and W alleles result in the formation of white flowers. If 2 purple flowered plants of genotypes capital P small p, capital W small w are crossed, then the expected phenotypic ratio of offspring would be. So I have made the cross here, I have drawn the Punnett square, and I have counted the number of purple and white flowers, and I got 9 purple and 7 white. So the answer is option C. You can go through the Punnett square. Then, autoclaving is the method of sterilization of glassware, certain buffers and media. Usually at what temperature and pressure it is used in the laboratory. It is 121 degrees Celsius and 15 pounds of Psi respectively. Answer is option B. The next one. Agrobacterium genetically transforms the plant roots and produces crown galls. The major genes involved in this process are, so they are Vir and tDNA. So the answer is option B. The next one. The genetic material in general is DNA in majority of the cases of evolution. However, single-stranded RNA, that is negative sense, positive sense or ambisense, double-stranded RNA, single-stranded DNA and double-stranded DNA in the form of linear and circular modes are available in these organisms. So they have told that you can find single-stranded RNA of negative sense, positive sense, ambisense, or the genetic material might be a double-stranded RNA or a single-stranded DNA or a double-stranded DNA. And these can occur in the form of either linear forms or circular modes. So these kind of genetic material are found in which organism out of the following options? So it is the viruses that carry these different kinds of genetic material. So answer is option C. Then next one. Which one of the following types of bonds is least likely to stabilize the three-dimensional folding of most proteins? So out of the given, that is disulfide bond, hydrogen bond, electrostatic bonds, only ester bonds are least likely to stabilize the three-dimensional structure of folding of most proteins. So answer is option C, that is ester bonds. Now next one. Which is not involved in the processing of mRNA precursors in eukaryotic cells? So capping at 5' ends, that is there for processing mRNA. Then poly A tailing is also there. Then excision of introns is also there. So they have asked for the one which is not involved in the processing of mRNA precursors. So transport of pre-mRNA to cytosol does not take place. It's not involved in the processing of mRNA precursors. Then next one. When the guard cells have a dash water pressure, they become dash and close the stomata. So when the guard cell have high water pressure, they become flaccid and close the stomata. So answer is option D. Then next one. If the frequency for the allele is 0.4, the recessive allele is 0.4, then what is the frequency of the homozygous dominant individual? So they have given the frequency of the recessive allele, that is Q is equal to 0.4. Then they have asked for frequency of the homozygous dominant individual. So P is equal to 1 minus 0.4, that is 0.6. Then frequency of homozygous dominant allele, that is P square, because you have two genome alleles, that is capital A and small a. So 0.6, the whole square will be 0.36. So the answer is option B, that is 0.36. Now I have seen that in most of the question papers, not only of Hyderabad University but other university of also, Hardy-Weinberg question is always there. You will get one question from Hardy-Weinberg. So be prepared thoroughly with Hardy-Weinberg problems. Then, which of the following statements about cysteine is correct? So cysteine forms when the CH2SHR group is oxidized to form a CH2SSCH2 disulfide bridge between two cysteines. So that is correct. So option A itself is correct, whereas other options are not correct. Okay. Next one. In plant cell, dash divide the cytoplasm into two cells. So it is the cell plate that divides the cytoplasm into two cells in the case of plant cells. Then crossing over generally occurs in dash during meiosis. So it occurs during the prophase 1 of meiosis. Crossing over take place during the prophase 1 during meiosis. So answer is option. Then which of the following is considered a macronutrient for most plants? So it is magnesium that is considered a macronutrient for most plants. Then a part of the biosphere that absorbs more carbon dioxide than it releases is called. It is called as the carbon sink. So answer is option A. Then next one. Which of the following statement is true? Shoot apical meristem is indeterminate whereas floral meristem is determinate. Shoot apical meristem is determinate whereas floral meristem is indeterminate. Both shoot and apical meristem and floral meristem are indeterminate. Both shoot apical meristem and floral meristem are determinate. So, the cells of the shoot and root apical meristem divide rapidly and are indeterminate, which means that they are not designed for any specific end goal. The shoot apical meristem, they give rise to leaves, flowers, while the root apical meristem produces further root growth. So the shoot apical meristem is indeterminate whereas floral meristem is determinate, because floral meristem has been designed for a specific end goal, that is the production of flowers. With that the meristem will stop dividing once the flowers are formed. So shoot apical meristem, it is indeterminate. It gives rise to leaves and flowers. And for floral meristem is determinate. So the answer is option A. The next one. Pineapple has 13 spirals in one direction and 8 in the other. These numbers are successors in dash sequence. So they are successors in the Fibonacci sequence. Answer is option B. Stomata are found in all shoot organs except. So stomata are found in all shoot organs except petals and stamen and filaments. So answer is option C, that is petals. Then plant cells are omnipotent, totipotent, quasipotent, pluripotent. So they are totipotent. They give rise to a whole organism from one single cell. Then next one. Frame shift mutation often arise because of insertion. Option B. If you insert a specific nucleotide, then it will result in frame shift, shifting the frame. So it results in frame shift mutation. Then in size exclusion chromatography, molecules that are dash move around the beads in the matrix and therefore move through the column dash. So in size exclusion chromatography, the molecules are separated based on their size. So the molecules that are larger size move around the beads in the matrix. Matrix is the chromatographic matrix, and therefore move through the column at a faster rate or quickly. So the answer is option D. Red rot disease of sugarcane is caused by. So it is caused by Colletotrichum falcatum. Answer is option A. Groundnuts, cotton seeds, etc. are aflatoxins. These are produced by. So these are produced by Aspergillus flavus. Answer is option B. Then in heterosis, dash can be observed. So in heterosis, superiority of the heterozygote over either homozygotes can be observed. So answer is option B. Then caraway, cumin, fennel, asafoetida are commonly used species in Indian cooking, and they are well known for their medicinal properties. They belong to the family. So all these, that is caraway, cumin, fennel, asafoetida, they belong to the family Umbelliferaceae. So answer is Umbelliferae. Answer is option A. In contrast to bacteria, eukaryotic chromosome need multiple DNA replication origin because their replication rate is much slower and it would take too long with only a single origin per chromosome. So eukaryotic chromosomal DNA, they have got multiple origin of replication. So that is replication starts from different origins. Whereas bacteria have got only a single replication origin. So why? Because the eukaryotic chromosome, if it is having only a single origin of replication, then the replication rate is lower and it would take very long time to replicate one chromosome. So answer is option D. Then which of the following statement is true? A photon of blue light carries more energy than photon of red light. A photon of blue light carries less energy than photon of red light. A photon of red light carries more energy than photon of blue light. Photon of both blue and red light carry equal energy. So red light has longer wavelength, a smaller frequency, and thus the photon energy which is smaller than the blue light. So photon of blue light carry more energy than photon of red light. Answer A is correct. The transcription of DNA to messenger RNA occurs. So it occurs in the nucleus. Answer is option C. Which of the following statements is not true about X-linked recessive characters in human beings? So answer is X-linked recessive characters. More males than males are affected. If a female has the characteristic, all her sons will show it. That is also correct. Females can be carriers of the gene without showing it. That is also correct. All sons of an affected male will show the phenotype under study. That is not correct. So they have asked for the one which is not true. So the option D is not true. The next one. Penicillin and synthetic derivatives are often used to treat bacterial infections. What is the target of such penicillins in bacteria? So penicillin inhibits cell wall synthesis in bacteria. So answer is option B. Transcription termination in bacteria is either dependent or independent, mostly based on intrinsic factors. Of this factor, so transcription termination is either dependent or independent and is dependent or involves which of these factors? So it is the rho factor that is involved. So answer is option D. Inhibition of folic acid biosynthesis in bacteria is achieved by the use of a synthetic antibiotic that binds to dihydrofolate reductase. Identify the compound. So it is trimethoprim, that is the compound. Then Drosophila flies of genotype X0 would be. So they would be sterile males. Answer is option A. Then next one. Identify the corresponding target site for the following restriction endonucleases. For ECO R1 it is GAATTC, so L3. So you can delete option, not delete, but you do not have to consider the option B. That is L5, that is wrong. Then BAM H1 is GGATCC. So option D will be the correct answer. But we will see for the others also. Then hint 3 is AAGCTT and SMA1 is CCCGG. So answer is option D, that is L3 M1 N5 O2. Now next one. A bacterial cell divides once every minute. It takes these dividing cells an hour to fill a glass. How much time is needed to fill half of the glass? So the answer, this is a usual question not only for MSc entrance examination, but for most of the different entrance examination this question has been asked. So in the life cycle of bacterial cell is 1 minute. It means that the number of bacterial cell will divide in 60 seconds. So if it takes 60 minutes to fill the cup, it must take 59 minutes to fill half the cup, because the cell will divide in the last 1 minute. The cell will divide to fill the cup, fill the full cup. Okay, so in the last 1 minute the cell will divide into 2 to fill the full cup. Hence 59 minute is the answer. Answer is option D. Then a technique for identifying individual chromosomes in a cell is, so it is by Giemsa staining or by G banding. Answer is option D. G banding or Giemsa banding is a technique used in cytogenetics to produce a visible karyotype by staining condensed chromosomes. So answer is option D. Which of the following depicts the 3-point test cross? So a 3-point test cross is used to determine the loci of 3 genes in an organism's genome. An individual heterozygous for 3 mutation is crossed with a homozygous recessive individual and the phenotype of the progeny are scored. So it should be a heterozygous individual with a homozygous recessive. So out of these, option C is the correct answer. You have got the heterozygous individual crossed to a homozygous recessive. So that will depict a 3-point test cross. So that's it. Please like, share and subscribe. Thank you for watching my channel.